So I'm going to talk about a variety of research, sort of very closely coupled research efforts that have to do and that rely on the integration of genetic, genomic, pathology, radiology information, and to some degree the motivation is to describe the actual research findings, but probably to a greater degree it's to talk about some of the methodologies and some of the approaches that I think are going to be important in the next number of years in terms of really applying biomedical informatics, leveraging biomedical informatics to understand development of treatments and evaluation of treatments in medicine. And so what I'm going to cover involves development of characterization at multiple spatial levels using pathology and radiology of cancer for the most part, and integration of that information with molecular data. And the motivations for doing this vary by the disease, but I'm going to be talking about cancer and really primarily about brain tumors, although I'll be talking about a little bit some results involving a neuroblastoma, and I'll also be talking about issues having to do with characterization of type of disease and clinical phenotype. And I debated actually whether to include analyses having to do with healthcare data and backing into clinical characteristics from that, decided that that actually might be of general interest. So that's slightly disconnected from some of the rest of the work that I'll be talking about, but I think plays into it pretty well. So I'm going to talk then about in particular, a set of new glioblastoma multiforme, new GBM subtypes that we have discovered through analysis of whole slide images. And as you'll see, the subclassification of GBMs, and I'm sure most people know this already, is a very complex, confusing thing. So we've added to the confusion. I don't think we've really clarified a whole lot, but we now have yet new ways of characterizing GBMs. And then I'm going to talk about this in the context of characterization of GBM tumor microenvironment. The hope with GBMs and many types of cancer is that the increased degree of complexity that we are able to understand through analyses pertaining to systems biology and tumor microenvironment may, in the hopefully not too distant future, lead to a degree of conceptual clarification of the mechanisms and potential modes of treatment of some of these very difficult diseases. I'm going to talk about pathology data analytic tools and techniques that were motivated by this work and are potentially and actually are applicable to a variety of other situations, both in research and healthcare. And then, as I said, I'm going to talk about electronic health record extraction and characterization of disease types from that sort of information. So our in silico Center for Brain Tumor Research, which was funded 3 years ago, joint with Dan Bratt. So Dan Bratt and I run this thing, and we've had a number of follow-up grants from this, was dedicated to the concept of taking very comprehensive molecular information generated by the Cancer Genome Atlas and Rembrandt projects, and I'll say a few things about those, but and marrying that with both pathology and image data. So the concept really was that the understanding of disease has a strong pathology component. I guess I'd be expected to say that as I'm a pathologist, but really if you think about it, if I could tell you or somebody could tell you one thing about most cancers, the pathology classification would be perhaps in many cases the most or one of the most predictive things that you could learn in terms of guiding what sort of treatment or the ultimate prognosis and seriousness of the disease. So pathology data is extraordinarily important, and for the most part it's been dealt with in a very descriptive way. that in the last 15, 20 years, as we've moved further and further up towards high throughput and next generation sequence and characterization of gene expression and epigenetics, really for the most part, when one looks at a study, you've got all the molecular stuff which is analyzed with high throughput methods, and then you've got a human description from a pathologist saying, oh, and by the way, it was an oligodendroglioma, class 2 or some such. So the concept that one actually could and should use high throughput methods to, at the minimum, reduce inter-observer variability and hopefully actually also get better resolution in terms of subclassification, that one could use image analysis in a high throughput mechanism in pathology was a plausible notion, and the reviewers bought off on it. So we needed bioinformatics folks, we needed pathologists, we needed image analysts. We also included radiology in this group. So it's really a fun group. It's 20 or so people locally, and actually a larger group of collaborators, which includes Stanford University, Thomas Jefferson, Henry Ford Hospital. So again, the context here, the broader context here, which is I think really in the context of a talk like this is the most important, really is the general, what I'm going to talk about in GBMs as an instance in the more general concept of developing methodologies that take a global look at prediction of patient outcome in response to treatment using not only omic data of various sorts, but also using quantitative pathology and radiology features. And again, as I've described, really, when you actually look at any study, this is done already in the sense that a human will look at the tissue, and if imaging is used as a biomarker to describe advances or treatment response, it's used. But they tend to be very sort of loosely knit together with some high throughput analyses of the molecular stuff, and then generally a bunch of human descriptions involving the radiology and pathology, and then generally some statistics gluing it all together. So we're trying to make this sort of more reproducible and quantifiable. So a little bit of context about GBM. So GBM, as I'm sure you know, is a very rapidly fatal disease. Median survival is 60 weeks following standard treatment. Survival is 14 weeks following resection alone if there's no treatment. The lower grade tumors, survival is much longer, substantially longer, but once the tumor progresses to GBM, survival is short. So that's sort of heads up on GBM. If you look at pathology of GBM, and I'm not going to get into a huge amount of detail, but it's sort of useful to have some sort of idea as to what we're talking about here. You have pathology of the broader class of brain tumors. You've got astrocytoma type tumors, of which GBM is one, which sort of the parody is that the nuclear shape is a sort of a potato, and an oligodendroglioma, which is a related type of brain tumor where it's a sphere in a sort of a ball-looking thing. But of course, in real life, you have a lot in between. So one of the things that we started out with was attempting to distinguish between oligodendrogliomas and astrocytomas using a process where we would segment the nuclei and surrounding regions. And in a virtual slide, so a virtual slide is about 100,000 by 100,000 pixels. So you take a slide, you put it in a machine, you get this rather large set of pixels uncompressed. It's, call it 10, 20 gigabytes. And they're roughly about 100,000 cells that you get when you segment these things. So one process was to develop mechanisms of reliably segmenting these things and characterizing their shape, and I'll talk about that, and then using this characterization in order to link the characteristics of the cells to outcome and molecular characterization. Now, one of the things that's actually kind of interesting about this is, in addition to the sort of biological issues, you have a huge amount of data, you have a huge amount of features, and one of the, I think, important future aspects of this type of work is that I think as we move forward into the next 10 or 20 years. My belief is that what is going to happen increasingly as we move towards digital pathology is that we will be routinely extracting features and managing features in very large numbers of slides. So this is a lot of data, and I personally actually see quite a bit of clinical carryover to this. I'll talk about that a little later. Again, a few pictures of GBMs just to sort of fix ideas as to some of the stuff we'll be talking about. So as gliomas progress, so this is the GBM, and what you'll see is you've got areas of dead tissue, and this is something called a pseudopalisade. So you've got dead tissue in the middle, and then you've got this structure where you've got nuclei that sort of look like they're running away from the dead tissue, and then you have areas of new blood vessel growth that you don't actually see here. This is a lower grade tumor that doesn't have that stuff. When you look on MR with contrast, you can see the leakage of blood associated with these scrappy blood vessels. So the reason why this is actually relevant is you've got stuff going on at multiple scales. And when I got to Emory, knew Dan Bratt, who's my collaborator in this from Hopkins, actually. We were residents together. I was sort of a superannuated residence because I was a computer scientist for 10 years before. He's a neuropathologist who's been very interested in the systems biology of GBMs. And there are things going on at both the small scale, and I'm going to talk about the nuclear classification, but there are also things going on that relate to the systems biology having to do with the relationship between factors such as HIF-1 alpha associated with the dead tissue, the generation of angiogenesis, possibly cancer stem cells and the like. So there's really a lot going on here, both at multiple scales, and different parts of the story can be seen at multiple scales. So switching context for a minute to gene expression. Okay, so if you take a tumor and you assume it's homogeneous and you grind up a specimen of it and you do a gene expression analysis, or you do a collection of patients, it's been found that depending on how you cluster, there are a number of gene expression categories, and generally the field has come to the opinion that there are 4 categories, although some people might argue that there are 3. And they actually have somewhat different systems biology characteristics. There's a paper that came out in Nature 2-3 years ago describing Cancer Genome Atlas GBM pathways. I'm not going to go into it, but it's been reasonably well characterized from the standpoint of both gene expression and genetics. And the different categories have different systems biology characteristics, as well as to varying degrees different expectations for survival. So there's already been work done associated with this classification. This classification came from Verhoek in a paper a few years ago, but it's been replicated in different ways. So one of the things we did early on in the study was to say, well, okay, let's look at the relative importance of the gene expression classification and how that relates to the pathology classification. So what we have here, these different colors represent the different classes of gene expression, with the proneural being the good one, the mesenchymal being the bad one, the other ones being in between. And what you can see is what you'd expect, but I wanted to emphasize that the pathology classification, GBM versus astrocytoma versus oligodendroglioma, are very important, as are the gene expression class. So the outcome, the outcome is a function of both of these. And we did an analysis of some of the public Rembrandt data sets. People had actually at that point only looked at the gene expression classes in GBM. So far this doesn't involve any image analysis, but there's a plausibility argument that in fact a combination of the gene expression and the pathology is important. The data sets which I alluded to that we were dealing with involve something called Rembrandt and the Cancer Genome Atlas. How many people have heard of Cancer Genome Atlas? Raise your hand. So a few people. Okay, so this is actually really, I think, an important thing, which I'll say a few things about, because again, from a methodology point of view, this is really critical. Okay, so about 5-6 years ago, and it really picked up in 2008 with ARA funding, the NCI decided that they would blow a few $100 million on a obsessive molecular characterization of different types of tumors, and they started out with GBMs. Okay, so this is the sort of proposal that would get limited enthusiasm from a study section, because the genetic analysis for each patient was about $100,000, and the hypotheses were a little scanty. So basically, enthusiasm is reduced by the investigators' relatively high price tag and the lack of specific aims. But it was a very cool thing, and in some sense, it's a little bit like CERN, although maybe a factor of 50 cheaper, in the sense that you have this very high throughput mechanism. The deal was that tissue was accrued from a bunch of hospitals, including Emory, and sent to a center to make sure that it's mostly actual tumor, and then slides are made, and then the tissue is aliquoted and sent off to a bunch of different centers that do all sorts of types of molecular analyses, and these centers were competed for with great vigor. So there's gene sequencing, there's DNA copy number analysis, there's gene expression. For a while there are 3 different platforms that were used. There's methylation, there's microRNA, and there's all sorts of stuff. So it was basically very much a discovery science approach where, when you think about it, if you think about your own R01 or program grant, you're not going to be able to just do a whole lot of different types of omic analysis just because. Right? I mean, so one of the problems is that if you go on, if you go to public sources and you download different complementary types of data, they're probably not going to come from the same patient, right? So this is very cool because you have a single patient and a single specimen under a controlled circumstance, and you get all of the stuff is complementary. It relates to the same specimen. So our approach, and there were a number of projects that involved in analyzing this data. So our approach was to do this analysis, but in the context of bringing in both the digital pathology information as well as neuroimaging information, and sort of integrate this together. So the reason why I think that this is important in a broader sense is when you think about the dramatic decrease in costs in all sorts of molecular studies, particular next-gen technologies, it's not $100,000 per patient to get the equivalent of what was done here. I actually probably should work it out what it would cost now. I haven't, but my guess it's maybe 1,000, maybe 500, because the early analyses weren't whole exome or whole genome. They're SNPs. I mean, they're doing more now, and so increasingly as next gen sequencing gets less expensive, your population translational studies can easily have this many different kinds of complementary types of data. When you consider how expensive it is to run a patient through any kind of a clinical study, the costs of doing the paperwork associated with data safety committees, adverse event recording, keeping track of just the whole management of the clinical situation, the relative cost of doing the omics analysis is a decreasing percentage. So there's no particular reason, and I think it's going to be increasingly common that studies that involve really almost any kind of disease will involve this sort of complementary information if it makes sense, which I think in many cases it will. So there's a bunch of, so we also gathered digital slides. Initially they were only weakly connected with this study. The tissue showed up, it was aliquoted, it was analyzed, slides were made, but it wasn't really exactly part of the program that they'd be scanned in. So the Henry Ford folks, Tom Mickelson and Lisa Scarpace, were wonderful because they made a lot of that their cases available, all of their cases available. We contributed our own cases. So some of our slides actually came from institutional sources. Increasingly, some of it came from TCGA and Rembrandt sources. So these are some of these. So we have roughly 1,000, 1,500, maybe like 1,500 slides. So the approach we took to analyzing this stuff is as follows. So if you take a patch of slide, one of the sort of drop dead obvious things to do is to look at the characteristics of the nucleus and surrounding cytoplasmic regions. If you're working with a neuropathologist such as Dan Pratt and you ask, okay, how are we going to distinguish one sort of glioma from another? The nuclei are definitely one of the number of things, but very important thing that one keys in on. And it's a relatively straightforward thing to segment nuclei. It requires a lot of tuning and validation, but it's certainly a task that's done by other folks and that we've done. And then you generate a whole bunch of features having to do with the area, with the energy, the entropy, the texture. And it's basically what's the shape of the thing. Is it grainy? Is it clumpy? You know, things along those lines. So what you have, and you can, one way I think of this sometimes is a solid state flow. Right? If you're familiar with flow cytometry, in some sense what you do is you extract these images and then you characterize their features. And if you're doing immunostains, and I'll say a few things about multiplex quantum dot, you can actually get a bunch of local molecular information. So it has, in my mind, a lot of relationship to flow, at least in a sort of high-level sense. But it's in tissue. We're not dissolving anything. It's sort of there. So as I mentioned, one of our first tasks, because Dan Bratt is a neuropathologist, and he makes the distinction between oligodendroglioma and astrocytomas, the question was whether GBMs that had some degree of oligoness, if some of the cells tended to look sort of more like oranges than like potatoes, did that actually make a difference in terms of outcome? The answer turns out to be no, although there do seem to be some molecular differences we found. But as an early exercise, we extracted these nuclei, and then Dan and his collaborators labeled a whole bunch of nuclei, several thousand, as to degree of oligo versus astroness, and then we fit it. So one of the questions that came up in sort of informal conversations before this is, well, how do you validate this stuff? And there are a bunch of different ways. I mean, one thing you can do is to make sure that your segmentations are actually segmenting nuclei by having pathologists, look at this. Another thing you can do is to take distinctions that are pretty well recognized and see if you can reproduce them. So, but then if you want to be a little more ambitious, and one of the things that we weren't really sure whether to expect, and so we were initially actually thinking that we would try to formalize the distinction between oligodendrogliomas, basically that there was this intermediate type of brain tumor that is GBM but is sort of oligo-like, and the question was, were there differences in outcome and were there molecular differences? And the answer seems to be, well, there might be differences in outcome, but we couldn't really, our N isn't large enough to detect them. There do seem to be molecular differences. But it turned out actually that even within the collection of GBMs, what we ended up doing, and I'll say some things about it, was to do an unsupervised clustering. So we basically segmented the nuclei and extracted features and did an unsupervised clustering, and actually, as I'll talk about, discovered that there appear to be both outcome and molecular differences between these clusters. So in other words, if you look at the population of nuclei in patients, that there are substantial differences between classes of GBMs that are distinguishable, reasonably well separated, and do appear to have some significance. So what we did is we took 200 million nuclei from 480 slides corresponding to 167 distinct patients in the TCGA data set, and we used a k-means consensus clustering method. So we basically used a bunch of different starting points, did an unsupervised set of unsupervised k-means clustering in order to, and we looked at varying numbers of clusters, and basically determined that, and there's nothing sacred about 3, but it appeared between 3 and 4 clusters appeared to be sort of the right number in terms of separating these clusters. And then what we did is to look at the outcome of these clusters. So what we did not do, which is something we were tempted to do but we didn't do, is one thing you can do with a relatively small data set is to say, well, there are differences in outcomes. Once we know the differences in outcomes, can we find out how to cluster it so as to be consistent with the differences in outcomes? Right? That would be bad. Right? No. So what we did, I mean, you can get away with that sometimes, but you got a lot of explaining to do when you do that sort of thing. So what we did instead was to do a consensus clustering without regard to what the outcome was, or to really anything except for the segmented nuclei. And then once having generated candidate clusters, we then said, okay, what do we make of these things? What are the differences in outcome and what are the differences in molecular characteristics? And then the other thing that we did was, as I'll talk about a little bit, is to validate that with another data set that was unrelated to this data set. So there appear to be three clusters here, and one sort of question you can ask is, well, how does that relate to the gene expression characteristics? And the answer seems to be to some degree. Interestingly enough, these gene expression classes, well, interesting to us if you play with this sort of thing, maybe not to other people. In this particular subset of the TCGA data, didn't actually significantly explain outcome, whereas our morphology clustering did. But then if you look at the different morphology clusterings in terms of the gene expression, you can see there do appear to be some systematic differences, but certainly there's a huge overlap. Just to give an idea as to what these nuclei look like, we generated representative nuclei for each cluster, did a validation with 84 GBMs from our Henry Ford collaborators, so these were separate patients, and found clear separation, at least between the two, the worst cluster and the best cluster, not as good a separation with the middle cluster, and then went through the sort of usual bioinformatic gene expression, gene enrichment analysis sort of thing, where we sort of systematically said, so we actually discovered this stuff. We were all a little skeptical, especially Dan Brett, who's a neuropathologist. It's like, he said, this is very cool, but how do we explain it? So we looked at it and we ran it again, and he said, well, what do we make of this? How do we justify it? We put out a paper in a Transactions of Biomedical Engineering, about the methodology, mentioned the results, and but basically we felt that we really needed to try to come to some understanding as to what was going on, and so Lee Cooper and Carlos Marino are the two people who sort of headed up the bioinformatic analysis of this, sort of systematically went through each cluster to try to find out ways in which they were different. So I mentioned the difference in prognosis. There are also differences in terms of hypermethylation. The bad one, you've got a lot of genes hypermethylated. Some differences in signaling pathways. Interestingly enough, and actually we've got some work looking at lymphocytes-associated tumor-infiltrating lymphocytes. This is lymphocyte enriched, and that does appear to be sort of independently important. So we have this. So we have this characterization. It's rather detailed. It's interesting. It's not all entirely clear what it means, but these appear to be reasonably robust. Interestingly, Joe Gray came up with rather similar results in a sort of preliminary paper, which made us decide, okay, we should probably get this thing out. So we're using the same data, but somewhat different, but basically the same type of methodology. So that's sort of a high-level overview of the use of nuclear morphology to subclassify. Where we're going with this, and I'm going to be talking more about methodologies and some other systems biology issues, but where we're going with this is really developing a large-scale pipeline to do this for a larger number of brain tumors as well as lung and other types of tumors. So I'm going to say a few things about requirements associated with this. So you've got 10 to the 10th pixels for each whole slide image. So the workflow of this thing is you have a pathology slide and you put it in a machine and you scan it in. And when we got started with this stuff at Hopkins in the mid-90s, we used a robotic microscope that took half a day to generate a whole slide image. It was 10,000 photomicrographs that we tiled. And we actually published that in the AMIA conference, the annual AMIA conference, and it seemed sort of cool. And I gave grand rounds at Hopkins, and I said it'll be 10, 20 years before anybody actually uses this stuff, and it was true. It's 10, 20 years. Anyway, but now you can actually do a slide in a few minutes. If you believe some of the vendors, you can get a high-quality image faster than that at high power. So these things have gone industrial scale, mostly used for research, but there are a few places like Toronto, University of Toronto, that are actually going live with pathology, digital pathology, and using this as, for actual clinical use. There are a number of whole slide images per patient. You've got quite a lot of features per whole slide image, because the approach here is not just to digitize the slide and use that as sort of a nice high-res mechanism for a human to look at. The approach is to extract a zillion features and then try to do high-throughput feature analysis to augment what humans do in order to find better subclassifications and reduce inter-observer variability. So a really critical point in terms of actually making some sense out of this stuff, and one of my students, Tony Pan, and collaborators Tassin Kirk, is working very closely with Oak Ridge National Labs on this is a driving model for large-scale data analytics, is when you have a cascade of algorithms and you come up with findings of the sort that I talked about, there's a natural question of, well, how sensitive is this on how you segmented your nuclei? How sensitive is it on your choice of features? There are lots and lots of variables in such things. So uncertainty quantification and sensitivity analysis is really important. Now, I think we did okay with the GBMs because it took us a couple of years, and we had there was kind of a community chewing on this, and we had some semi-competitors who were also doing the same thing, and they came up with a similar thing. But if you're going to turn this into a high-throughput thing, you need to be more systematic. So in my view, you really need to be able to do very systematic and quite expensive sensitivity analyses comparing different algorithms and different parameters in algorithms. And that's actually one of our research areas, joint with computational science group at Georgia Tech. So that's going on. And I'll say some things about the HPC aspects of this, where one does this. Another feature is, and I didn't get into the details, but the way we did the initial phase of the analysis that I talked about is we did segmentation and generation of features on a patient-by-patient basis. So we did electronically circle several 100,000 nuclei per patient, but then we aggregated them into a data descriptor so that there are 74 feature characteristics, and we had an average and variance of each feature characteristics on a patient-by-patient basis. So we were basically looking at kind of the eigenfeature per patient, which makes sense in GBMs if you're not imaging the necrotic areas and you've got sort of lots of cells that are that are not that dramatically different from one field to another. But really, you should be doing things on a finer grained level that is much more expensive, especially when it comes to the clustering. So that's work we're doing in the Oak Ridge context, where we're basically actually doing k-means consensus clustering at a cell level. So you're actually doing clustering problems with hundreds of millions or billions of objects. So the general concept then of squeezing information from spatial data sets, so this is my orange juice squeezer, I think is sort of a general model that is going to be increasingly important. So getting back to where I started out with, we were talking about integrating pathology and radiology information with omic information. In some sense, what you're trying to do, for those of you who are computer science types, you could think of it as being sort of the IP, internet protocol for sort of imaging stuff, rather than sort of having these vague analog quantitative descriptions. What you try to do is to break these images down into large numbers of hopefully somewhat meaningful chunks of features, and then use the features along with the molecular information to ask questions. So the management of the features is a rather involved business, and Fusheng Wang, who's in my department, who's a database guy who had actually worked at Siemens before that. He's a PhD in the database area. Has developed a number of tools, some involving spatial GIS databases using IBM DB2 with spatial extensions, and then basically worked with the team to develop a way of representing components of pathology images in a formalism that we call PACE, pathology image archive standards or some such, and then using that to support queries, just as if one had a geographic information system. If you want to know how many houses are within 2 miles of 95, or how many buildings that are more than 5 stories are within a mile and a half of a mall of greater than a certain size. A lot of the questions that one wants to ask in the context of pathology image analysis can be put in a context of that sort, whether you're talking about matching, how consistent are methods for segmenting objects. So if you have 2 different algorithms that are segmenting nuclei, how different are they? If you want to ask questions about how cells that might have a certain immunostain are, how many of them are their characteristics close to blood vessels, then you get queries of that sort. So his tools handle that sort of thing. The standard, well, it's not exactly a standard because we've just proposed it. It'd have to be adopted to be a standard, but we've published on it, and I think something along these lines, probably not exactly this, will be used. And this was actually done as part of CA BIG as well as with R01 funding to describe the features of the pathology image. And this is sort of, in some sense, harks back to BIRADS, if people are familiar with that in the radiology domain, where mammography images features are described. And then he's developed an infrastructure. The group's actually developed an infrastructure where basically when one does the analyses, you load the images into this data management system, and then can do spatial queries to look at, say, differences between segmented nuclei, or to extract a patient-wide feature vectors of the sort that I've talked about. So basically what we're talking about is a data management and query system for handling the zillions of features that come from these sorts of analyses. So obviously this is nothing specific. There's nothing specific about this to brain, and rather similar sorts of methods seem applicable to radiology, although our main focus in this particular software development effort has been brain. We also have a Hadoop version of some of this stuff that I won't go into a whole lot. This is computationally intensive, and we have a group of folks who have been working on optimizing this. If people are familiar, there's something called the fire hose pipeline at the Broad. Okay, the basic concept of that, and where we're aiming, is that as one incrementally gets new information, or one wants to try new algorithms, one can rerun the analyses and then ask, okay, what new things have we found out? So it's very expensive to do this, and there's been a number of our folks have developed tools and code that map the pipeline that I described to a platform that contains a combination of CPUs and GPUs, as well as actually other platforms. But we've focused a lot of our benchmarking efforts on that, and obtained the appropriate impressive speed-ups from this platform. So I'm going to switch contexts a little bit to similar methods, but a different problem, and especially since I'm visiting a children's hospital, it seemed important to have a children's disease, especially since we worked with Hiro Shimada on an analogous problem with neuroblastoma. Okay, and Hiro Shimada is the god of neuroblastoma pathology classification. He's at LA Children's, and by a striking coincidence, he is the person who is the regular user of the Shimada classification, and at least at the time we were working with him 3-4 years ago, was reading all of the slides from North America and I think Australia as well. So this was an easy problem in terms of inter-observer variability, because there was only one observer. It was Hiro Shimada. And so we went through the process of attempting to build an electronic Shimada interpreter. And Jun Kong, who's actually now a research scientist with us at Emory, he was a grad student then, and Metin Gurkin and I worked with Hiro for a number of years, and systematically went through, and I won't plague you with the classification of neuroblastoma, but it's a moderately elaborate classification where you're sort of looking for different sorts of features, some of them cellular, some of them having to do with little fibrils and fibrous tissue. And so there are a bunch of things you're looking for, and there's a bunch of classifications. So basically went through in a sequence of steps and replicated much of what, really most of what he did. And it was an interesting process, and I think we learned a lot, which was sort of the intent in the sense that what he said he wanted out of this was that he wanted to someday not have to read neuroblastoma, but he didn't trust anybody else to do it, so he thought that having an electronic system would be useful. And Metin Gurkin has picked most of this stuff up and continues to work with him. And I'm not quite sure what the stage of the implementation was, but I think it was an interesting case in that it at least demonstrated to us and him, and we published a bunch of papers, that you could do a reasonable job of this. And people in the meantime have done analogous work with prostate cancer and a number of other types of cancer, demonstrating that if you prepare and process the tissue in a standard way, which is a critical point, and you're not looking for zebras. In other words, if you're trying to get a, say, 95, 99% concordance, then you can generally do pretty well with a sort of cascade of image analysis methods in pathology. Okay, so that leads to sort of a meta question, which is, well, why do you want to do this, right? And this is always a sort of thing about technology, is that sometimes it's like, it'd be cool to do it. Why do you want to do it? Well, okay, so I see two reasons to want to do it. The real reason to want to do it is, I think, what we're doing in the In Silico project, which is to find new ways of classifying cancer that might actually have prognostic or hopefully treatment significance. The other reason you'd want to do it is to reduce inter-observer variability. I don't think the reason to do it is to get rid of pathologists. I wouldn't go for that. But I mean, also, when you really get down to it, people, fortunately, don't really have that many cancer slides to read. I mean, you're not really reducing that much cost from the healthcare system. But there is a lot of inter-observer variability amongst pathologists. But I think the more interesting reason is actually new science. But it is interesting. I think it's also notable as a sort of plausibility. If basically you can imitate a human, pretty well, then it's much more plausible, in my view, that you can look for new classifications. Because if you can't imitate what a human does, then I'd be a little bit more skeptical. I'll skip the adaptivity stuff. And we didn't agree with them 100%, but this was one dimension of the classification. But generally what we saw were distinctions that were kind of nearest neighbor, undifferentiating to partially differentiating. We might not completely agree, but there wasn't a lot of, this is totally different. But as is true with any computer algorithm, you can always fool it, right? So it is very easy if you train the thing on 3 micron slides and you give it a 5 micron slide, or you have a new process of staining, then it's going to totally mess it up. So I'm not arguing that magic is occurring. What I'm arguing is that in a well-controlled experimental setting with a consistent pathologist, if the wind is behind you, you can sort of do okay, which is pretty much what the radiologists have found as well. And I think that it's potentially important because it's the same thing with gene expression or next-gen sequencing. In the 90s, I was involved in some of the early gene expression work. I mean, not as a pioneer, but I just was at Hopkins, and I was working with some folks there. And the reproducibility was terrible, right? I mean, so any kind of a high-throughput method, has that issue. Well, okay, so I'm gonna now switch context a little bit and talk about some of the sort of larger scale issues involving radiology and larger scale pathology issues. And this work actually is done by an overlapping group of people. Adam Flanders from Thomas Jefferson, actually right here, is really the lead on this, although a number of folks from Emory have been playing really increasingly strong roles. Much of the work, and Carl Jaffe from the NCI has played a big role in this as well. And this basically involves a rather large number of radiologists who are actually using non-computer methods, well, they're computer methods, but non-image analysis methods, to manually mark up and characterize features of MR associated with brain tumors. Our guys are actually doing image analysis work as well, and just a huge group of people have been working on this. And then there's been a number of abstracts and a paper out with about 800,000 authors where we're sort of linking the characteristics of the imaging, the size, the shape, the position. Now we're looking at advanced imaging, diffusion tensor sort of thing, and looking at relationship between gene expression type, presence of new blood vessel hyperplasia in pathology slides, mutations, and so forth. So basically what we're doing is we're making connections between the molecular, the pathology, the radiology information, which oddly enough had not been done that much. I mean, one of the things that's striking about, and this is sort of halfway between basic science and clinical informatics in my view, the thing that's always amazed me about clinical informatics is how hard it is to do simple things. I mean, if you talk to a computer scientist about what's involved in EHR extraction, you know, I mean, at least until it became popular, it's like you've got a bunch of data. It's not that big of a data set, really, as data sets go. You swizzle around a little, and what's the big deal? I mean, I don't know why you're spending so much time on this. But then when you really try to do it, there's all sorts of issues. And it's the same thing with this stuff. When you actually try to make connections between the multiple types of genetic and omic data and pathology data and radiology data, you go through quite a lot of work to get some very high-level connections. So it's notable. So we've made these connections, and you know, the importance of them remains to be seen. Although I think they may be important, at least to some clinical extent, in the next phase, where some of the things that some of our folks are doing involves analyses to look at pseudoprogression in radiology imagery, in the sense that as you build up a sort of broader picture, you know, with multifactorial picture as to the nature of the brain tumor, the question is going to be, can you use these things in order to improve your prediction? Some work I don't have a lot of time to get into involving some analyses that have to do with systematic evaluation of percent necrosis on frozen sections and relating that to the TCGA genetic data by a variety of steps where we, in particular, Carlos Moreno played a really huge role in this, basically went through and defined some key transcription factors which appeared to be some of the same ones identified by the Columbia group as being master regulators. And so the basic issue here has to do with the scenario that the particularly, I mean, they're all bad types, but the worst type of GBM is the mesenchymal type, which tends to have a lot of necrosis. And when you look at the gene expression and the omic data, you tend to find certain pathways are particularly active, and there appear to be certain master regulators associated with those pathways. And when what we did was to look at a bunch of patient samples where we looked at not much necrosis to a lot of necrosis, and then looked at the changes in, as you went from the the patients with not much necrosis to the patients with a lot of necrosis, in what gene pathways were activated, it appeared to be the same ones. So finally, and I'll just have about 30 seconds for this, the phenotype, clinical phenotype characterization, and this does tend to be important in using institutional data for cancer studies, but it's important in many other things. We've done a bunch of work which actually, as I've talked offline to Peter and his group, appears to be actually quite complementary to some of the stuff being done here, which, to make a long story short, involves semantic ETL, mapping of data from electronic data records to derived data elements. And then we've used this to characterize in both the 180 hospital UHC consortium as well as in Emory Healthcare patterns of readmissions in hotspots, basically looking at different collections of comorbidities and readmissions and relationship between readmissions and hospital stay, geographic patterns of population. And again, these are really enabling tools. In general, one needs to understand both clinical phenotype, disease progression, and all of the molecular stuff. And in the future, I think in hospital and institutional studies, we'll want to do that. Ability to predict readmission rates using random forest methods. So, summary perspective to wrap this up. So we're interested in large-scale integrative data analytic methods to integrate clinical, molecular, pathology, radiology data. In the case of pathology and radiology data, image extraction, analysis, clustering are, I think, important things that we're doing a lot of work in. The issue of extracting and modeling clinical data is critical. Lots of people are working on that. And semantic ETL is our particular thing there. The goal is to characterize new cancer subtypes and biomarkers to predict outcome and treatment response. And really the importance of these sorts of methods, I think, ranges from basic science to getting a good hold of the relationship between genetics, genomics, and phenotype, and such things as comparative effectiveness research studies. So thanks to a very large group of collaborators in the integrative omics area, as well as our team of folks who do healthcare data analytics. And I want to thank you and happy to take questions.